I'm again here with my second portion of the same lecture in continuation of lecture 10, which is based on the single stranded RNA viruses of HCV. So in this portion of lecture 10, we will cover the remaining portion of HCV viruses. So uh, now let me start the HCV virus with its genotypes. So I already explained that there are more than 100 species of HCV which has been classified or categorized into different genotypes, and these genotypes were constructed or developed on the basis of its sequences available. So all the nucleotides were sequenced and they are categorized into different categories for the simple understanding or ICTV for rule criteria for demarcation. So only 7 genotypes has been made in HCV. All these genotypes shows variation from one group to another group up to 30 to 35%. So on other hand, each genotype is further split into different subtypes. This indicates that these subtypes belong to the same genotype. For example, genotype 1 has many versions, and all these versions are the subtype of genotype 1. For example, 1A, 1B, 1C, and so on. So the lower letter of English representing each case, each subtype of the same genotypes, and 1A is different on the basis of its nucleotide sequence, the whole genome. We are talking about the whole genome. Okay, so it shows variation 20 to 25%. Clear now? So if we are talking about genotypes, so each genotype is different from another genotype up to 30 to 35%, while subgenotype variation shows 20 to 25%. Clear now? So all these differences usually based on the basis of nucleotide availability. Subtype 1A and 1B are found worldwide and cause 60% of all cases. This indicates that 1A and 1B is more common in HCV patients. So already in the previous slide I was talking about these genotypes and subgenotypes of HCV. Here we can see that in the first column we can see the different genotypes 1, 2, 3, 4. Okay, and in the second column we can see the subgenotypes of, for example, if I'm talking about the genotype 1, so there are A to M. These are the subgenotypes of 1. So here the variation will be more between A1 and 2, 30 to 35%, while variation between A and B of the same genotype, it will be 20 to 25%. So here we can see these are all the genotypes of 2 and subgenotypes of 3 and subgenotypes of 4 and so on. If we are talking about the global importance or the global distribution or prevalence of HCV genotypes, so here we can see the worldwide distribution of all these genotypes 1A and 1B are highly common in all regions in America and Europe in Asian areas, while if we are talking here is Pakistan. So in Pakistan the most prevalent genotype of HCV is the 3A. In this slide we can see the prevalence of HCV. So the red zone show the high prevalence of HCV in regions. So it shows more than 10%, while the orange color representing the prevalence of HCV 5 to 10% in the regions. So here we can see the map of Pakistan. So here the prevalence of HCV virus lies between 1 and 2.4%. Now, before going the significance of HCV, so let me tell you why there are no vaccination for HCV virus. Because HCV infection is predominantly chronic and it has the ability to escape the immune system of the host pathogenesis. Because HCV is characterized by the rapid and permanent change in its antigenic structure. Antigenic structure changes occur multiple times per minute. Such antigenic variability of the virus make T lymphocyte and B lymphocytes unable to identify and respond to these permanently changed antigen, because once the antigens are changed, the body immune system, which is totally based on T lymphocytes and B lymphocyte. B is responsible for the plasma cell, which is responsible for the production of antibodies, and these antibodies are usually produced against the specific antigen. So when the antigens are changed, so definitely new antibodies will be produced. So it takes time. So the antigenic changes occur with a very high speed. That's why the immune system is unable to identify the new antigens. So if we compare the timing, so the rapid changes are more in HCV antigen as compared to production of antibodies. That's why the HCV is able to escape the immune system. So such antigenic variability of the virus make T and B lymphocytes unable to identify and respond to these permanently change occur. And one thing should be in our mind that these antigenic variation species are present in a single host or within a host is known as the quasi-species. Quasi-species are those species which show the antigenic variation, while the rest of region is the same. Is this clear? So if we are talking, so that's why we are unable to produce the proper vaccine against HCV because of these changes. So there are certain drugs which are still highly valuable for the control and used in a combination for the control of HCV. So now if we are talking about the significance of HCV, already we have discussed a lot about the HCV significance on the basis of its worldwide distribution and its different genotypes and its strands. So it basically causes a serious global health problem, because it affects more than 180 million people worldwide. 18 million people has been reported from Pakistan. So this is the basic shows the significance of HCV, because this is a liver disease, and from early in some patients it has been reported it is symptomatic, and in some people they are non-symptomatic, and in non-symptomatic we are unable to diagnose because the immune system is so strong, and sometimes it clears itself when the immune system is weak, or in older stages or in very early stages when the immune system is not properly developed. So there are certain chances that the virus from acute stage develop into chronic stage and finally develop into the carcinoma stage, which is responsible for the failure of the liver. Once the liver is damaged, or at this stage or at the last stage, which develop into hepatocellular carcinoma, there is no option, only transplant, and sometimes transplant is also failure. So that's why HCV is highly significant. Hundreds of thousands of people die each year from liver failure and liver cancer because of this disease. So still we are using certain drugs which can control the HCV virus, such as PI fusion proteins, because these proteins are responsible to discourage the attachment of virus to the host cell, because attachment is the most important part for the endocytosis, for the entry of the virus. And there are certain drugs which are responsible for protein inhibitors, proteases, responsible for further modification of polyproteins. And there are certain drugs which are used for the control for the control of the proteins, which is responsible for which is known as RNA dependent RNA polymerases, which is responsible for the development, for the construction of negative strand and play a vital role in the production of new positive strands in replication. So these drugs are still commonly used around the world. So this lecture, I'm going to wind up this lecture at this stage, and I hope so this all information will be quite enough. And if there is still any question, so anyone, all of you can communicate me through my email address.